So monohybrid inheritance is the first type of inheritance we're going to look at. There are several across the A-level course, but this one is the one that applies to the GCSE course. Normally in GCSE you would look at sex determination in the form of XX and XY chromosomes, but for A-level obviously we can bring in any characteristics. There's a number of key terminologies that will carry across from GCSE, and we will cover those to make sure everyone is at the same starting point. So to get us started, it's a simple question. The person on the left-hand side is the husband, the lady in the middle is the wife, and the person on the right-hand side is their neighbor. And the two people on the left have a baby, which is pictured at the bottom, thanks to the joys of Photoshop. And you're simply asked to give your opinion and advice to the husband because he's not convinced that that is his baby. What I'd like you to do is spend a minute, come up with what you think is the answer before progressing on to the next slide, and then we'll look at what the outcomes are. So there are a number of possible outcomes that you may come up with. The most common ones were ask for a divorce, leave the wife automatically because they're cheating on the husband, or ask for some kind of paternity test. If the husband was to pick one of those outcomes, then it doesn't end very well for him. However, if he does nothing because he loves and trusts his wife, it obviously ends much, much better. You may come up with some other outcomes. The most important thing here is to link the fact that what something physically looks like doesn't automatically relate to what its genetics is. So you have to be careful. There are hidden characteristics and things that appear and disappear depending on what's called epigenetics, and don't make assumptions about physical appearance and the genes that are causing that physical appearance. So there's a number of key terms that you have to use that you should have been introduced to at GCSE, may or may not have been, and you may or may not remember them. You must take the time to learn these off by heart now because you will not get through the A-level inheritance questions without using this terminology successfully. One of the words that's not listed here is the word gene. At GCSE, a gene is a section of DNA that codes for a particular characteristic. We do not use that terminology at A-level in that way. We have to say a gene is a section of DNA that codes for a particular polypeptide. When we go on to the term allele, alleles are all the different possible polypeptides that could exist for a particular gene. For example, if the gene was eye color, the alleles would refer to blue eyes, brown eyes, and green eyes because those are the different polypeptides that could be made in order to produce the different colored pigments in the eye. The terms phenotype and genotype are used to refer to the physical appearance of the organism, so what you actually see when you look at it, or the genotype refers to the alleles that are found in that particular organism's DNA for that particular gene. The term dominant and recessive refer to alleles. When you have a dominant allele, we write it as a capital letter, and when we have a recessive allele, we write it as a lowercase letter. A dominant allele is always seen in the phenotype, even if expressed with a recessive allele. So if you have two dominant alleles, or a dominant and a recessive, you will always have the same phenotype. If, however, you want to see the recessive phenotype, that only exists when you have an organism with both recessive alleles. We do have a situation called codominance, and codominance is when the two alleles are actually expressed equally in the phenotype. Quite often we find this in flowers and plants where you have red flowers and white flowers, and when you breed them you produce pink flowers where the white and red have blended together. The term heterozygous and homozygous are simply words to use when we want to talk about what alleles organisms have. If the organism is heterozygous, it means it has one dominant allele and one recessive allele. If, however, it's homozygous, it means both alleles are exactly the same. We say homozygous dominant if both alleles are capital letters, or homozygous recessive if both of the alleles are lowercase letters. So this is the standard inheritance family tree that you would see from GCSE. It's simply two organisms, one male and one female, breed together to produce one female offspring. If you look over in the key on the right hand side, you can see that squares and circles, squares represent males and circles represent females. Just be careful in the second one, it says healthy and infected. It's not the circle that's different between the two pictures. The difference in the two pictures is the fact that the infected is shaded in red, whereas the healthy is in white. And it's the red or white that determines if the organism is infected or healthy. It doesn't in any way say that this particular condition is linked through only females. It can equally affect males. It's just not applicable in the key. So just be careful, folks. Look at the difference between the two pictures in the key, and that is what they're using to represent individual characteristic traits. So as we said before, monohybrid inheritance is a simple pattern of inheritance carried across from GCSE, and a mono means one, so we're only looking at a single trait, a single gene. So GCSE, the most common type of monohybrid inheritance was the sex of an organism. If you are male and human, you have an X and a Y sex chromosome. If you are female, you have an X and an X. In this particular example, this family have had one older boy, then they had two girls, and they get pregnant with baby number 4. What is the probability, what is the percentage chance that they will have a boy? What is the percentage chance they will have a girl? What is most likely? Take a moment, think about an answer, and then progress. So when looking at patterns of inheritance, most common tool is what's called a Punnett square. And a Punnett square is just a particular arrangement that lets us show all the possible combinations of alleles and sex cells between two organisms to look at all the possible outcomes in terms of babies, and then work out the percentage of any particular characteristic being passed on depending on the alleles of the parent. In the previous example, we had one male and one female, the male having an X and a Y, the female having an X and an X. Each parent only passes on one of their two alleles. That means that the female will pass on the X on the left or the X on the right, whereas the male will pass on the X or a Y chromosome. And depending on which one is passed on by the male, the baby will be an XX, making it female, or an XY, making it male. If you can look at the diagram below, you can see that there's equal probability of a boy or girl being produced, even though they have had more girls in the past than boys. It is just as likely as baby number 4 being a girl as it is of baby number 4 being a boy. In the diagram below, we can see there are 3 different families. The families are separate because there's nothing linking them up. In the first family, we start with 2 parents who are healthy. Those 2 parents then reproduce and produce 2 female offspring. Remember, folks, it's important to use the word offspring because babies is a human-only term. We could be dealing with any organism, getting into the habit of using the word offspring. The 2 female offspring, one of them is infected, one of them is healthy. The infected female reproduces with a different infected male and produces an offspring that is male and also infected. The healthy female, number 3, reproduces with a healthy male, and they produce a male offspring that is also healthy. In the second family in the bottom left, we start with a male and female parent who are infected, and all of the offspring are also infected. But only one of the offspring is female. In the third family, we have a healthy male and a healthy female parent, and they reproduce and give us a female offspring that is also infected. So what I've got here is a way of identifying that when you're given one of these big diagrams with lots of families, and it says, how do you know if it's dominant or recessive? This is what you're looking for. You're looking for one of the offspring having the particular condition. As long as it's recessive, then you will have examples of both parents not having the condition while a child does. And it's simply because when you have the parents, if I go with the letter A's again, each parent has to have one of the dominant alleles so that they do not express in their phenotype the recessive condition. However, they must have had a second allele that is recessive, and those two recessive alleles have come together in the offspring, resulting in offsprings that express phenotypes that the parents do not express. And that's how we can identify if a characteristic is dominant or recessive. So what we've got is exactly the same arrangement as the previous slide. All I've done here is put in pictures of people rather than using diagrams to represent male, female, and if they have a particular disorder or not. In this case, the genetic disorder that's being passed on is if you are a wizard or not. And what you can see quite clearly there is that the original family 1 from the last slide, which is this family up here, we have 2 grandparents. We have one daughter who's infected with the disorder, in this case is a wizard, one daughter who is not a wizard. Again, they marry, and this person, Lily, marries James Potter, who's also a wizard, to produce Harry, whereas Petunia marries Vernon, and they produce Dudley, who is not a wizard and does not suffer from the disorder. The Weasley family are all a family of wizards. Hence, all of them have the condition and pass the condition on to their offspring. And then Hermione Granger, as we know, if you've read the books or watched the movies, has parents who are not wizards. And as a consequence, she has 2 healthy parents and produces offspring with the particular disorder. A couple of questions about the previous slide. What is so special about the Granger family? Well, the Granger family have an offspring that suffers from a particular disorder while both parents do not have the disorder in their phenotype. What is so special about Petunia and Dudley? Well, even though they are both phenotype of healthy, it's quite possible that they are heterozygous, in which case it is also possible that they could have offspring that will have the recessive phenotype depending on who their partner was. And again, that could appear in later books. Question 3, what type of genotype does a wizard have? Well, the genotype that a wizard has, using your keyword, should be homozygous because both are the same, and recessive because you need the lowercase letters. There's 2 ways that can happen. You can have 2 heterozygous parents, such as in the case of Hermione, where both her parents are healthy and she has a disorder, or you can have 2 parents that are wizards, in which case the offspring will get a recessive allele from each parent and will be recessive themselves in their phenotype. Question number 4 is for Harry Potter fans. What is a squib and what does it do to the pattern of inheritance? Somewhere along the series of books, J.K. Rowling decided that we should bring in what's called a squib. And a squib is where you had 2 wizards and the offspring is not a wizard. So you would have 2 homozygous recessive parents, and yet the offspring does not display the phenotype, which is the same as the parents. Now, what that means to the pattern of inheritance is, unfortunately, the books stop following monohybrid inheritance at that point, and it all gets a bit messy. But up until this point, we're going to ignore that and pretend it doesn't happen, because it means I can get all the Harry Potter characters in on the slide. As interesting as it would be to see an A-level exam question all about Harry Potter, it's extremely unlikely due to licensing laws. What you're more likely to see is the questions similar to what's in front of you now. When you get any question on inheritance, the first thing I'd always advise is identify your alleles and what the phenotypes are that are linked to each allele. So in this particular case, we have the letter A already chosen for us, and it says that it's a condition A is recessive. Therefore, we can say that the capital A means you're going to be healthy. And if you have lowercase a, then you will suffer from condition A. If we then say that the parents are heterozygous, we know that all the parents must have 1 uppercase and 1 lowercase letter. It then says rat X does not have condition A. For rat X to not have condition A, rat X must be 1 upper and 1 lowercase, your heterozygous condition, or it must be the homozygous dominant condition. We now have to use our knowledge of Punnett squares and monohybrid inheritance to identify a method for determining the genotype of rat X. So take a moment now and have a think about how you might do that. So we've had to work out if a offspring is homozygous dominant or heterozygous. And the method we use to work this out is called a back cross. A back cross is where we breed an organism with the recessive phenotype, because if it's the recessive phenotype, we know it has two of the recessive alleles. So if we do that, we know straight away that the recessive organism, its phenotype means that it has two little a's, which means it suffers from condition A. We have two possible outcomes for the offspring. The offspring could be heterozygous, or the offspring could be homozygous dominant. That was all the possible outcomes from the parent breeding last time. If we have a homozygous dominant offspring from the previous cross, when we breed it with the recessive phenotype, all of the offspring from those two parents will come in as heterozygous, which means that they will all look exactly the same. And in this particular case, that means they would not have condition A, whatever that happens to be. If, however, the offspring that was produced in the previous breeding, they would be one of two possible combinations. They would be heterozygous, which means they would not have condition A, or they would be homozygous recessive, which means they would have condition A. So what you would have to do in order to work out if the previous organism was the heterozygous or the homozygous dominant is breed it a lot of times with the recessive organism and then look at the offspring. You have to breed in large numbers, because if you only do one breed, by pure random chance, we could have had one of the two top combinations where the offspring are heterozygous. We will already get a definite homozygous recessive if we do enough breeds. So we have to make sure we're breeding large numbers. Hence we use things like mice and insects, where they will breed very large numbers in a very short space of time. So when we do a back cross, going back to our original question, we can do a back cross and find that both parents, even though they've got dark hair, actually are heterozygous. That means they have one dominant, one recessive allele. If they've got one dominant, one recessive allele, when we do the cross, we cross a Punnett square with two heterozygous parents. So one upper letter, one lowercase letter. And you should find that the possible outcomes are, one, we have homozygous dominant. Two of the outcomes should be heterozygous, and one of the outcomes should be homozygous recessive, and the homozygous recessive will give you that blonde child. So there should be a 25% chance of that having another baby and that baby being blonde.